Thank you. Thank you, Sandro. And thank you for your kind introduction. And ladies and gentlemen, it is really a pleasure being with you today here in Sorrento and in this very, very fascinating program. And I'm happy to be contributing. So I would like to start by also thanking the organizers, Professor Alvigi, Professor Peter Humaidan, Robert Fisher, and also Sandro Esteves, and to everyone who's been involved in inviting me here. I was given this topic, which I must confess, it raised an eyebrow when I received it, because I'm not particularly, I'm not a geneticist, I'm not particularly known for my work in the field of genetics. However, what I potentially thought the organizers wanted is, a more epidemiological, critical review on the evidence. And initially, I was quite skeptical on whether I should even accept to talk on the topic. But I must confess that after going into the literature and deep diving, I'm far more encouraged to what I was before I actually started this endeavor. So I would like to start with my disclosures. And we'll jump straight to this slide, which you've all seen and you all know. And, in different forms and variations, this is what virus stimulation does. And obviously, gonadotrophins is a key important element of ovarian stimulation for most protocols. But I think most of us agree that in 2025, ovarian stimulation represents an integral part of every IVF cycle. It doesn't matter where you do freeze-all, whether you do whatever you might be doing, ovarian stimulation is something you should strongly be considering. And what is ovarian stimulation? This is again the slide you've seen from Bart Fauser's group. Essentially, we're trying to we're trying to bypass the natural monofollicular selection mechanisms that nature has imposed on humans for good reason, and trying to save as many from the follicles that otherwise would end up in programmed cellular death or atresia, because we've seen that actually these follicles can have equal or even sometimes better quality eggs compared to what was destined to be the dominant one. So what is the aim of ovarian stimulation? Well, very simply put, is to obtain a good number of good quality eggs. And why a good number? And why not just one or two? Well, because it's well known, and many groups, including our own, has shown that getting a good number of eggs, meaning that you'll end up getting a good number of embryos, you'll end up getting more euploid embryos either day 3 or day 5, and many groups from Australia, from the UK with Sankara, from the HFEA, from the SART data as well, have shown consistently that observational studies support that when you get a good number of oocytes, you actually increase substantially your chances of a cumulative live birth rate. At the same time, we're all very weary of, let's say, excessive or the two extremes of this type of response. So we don't want to see a poor ovarian response, and has already been touched on by the two previous speakers, because we know it's associated with disappointingly low pregnancy rates and can be quite a bit of a challenge to manage. And at the same time, we know that getting too many follicles and too many eggs is, very strongly associated with an iatrogenic complication that thankfully many of us almost seem to have forgotten, which is severe OHSS. And for this reason, since, probably since 30 years, many colleagues started trying to produce the best way to predict ovarian response. And because potentially predicting the exact number might have been too ambitious, they said, can we at least try and predict the two extremes? So many different markers have been suggested. I think there is a consensus now that AMH and AFC seem to represent the two most valid and valuable markers, and we use them in everyday practice. So this is the latest meta-analysis, and you can see that we actually, if we pull all the studies together, both AMH and AFC seem to have pretty good diagnostic accuracy characteristics, as I call it, good predictive capacity with good areas under the curve, good and reasonable sensitivities and specificities, and positive and negative predictive values. And the same applies for low response. So if you ask any general reproductive medicine specialist, they would say that we rely exclusively on AMH and AFC, and that might be for good measure. Many colleagues, Antonio Lamarca and Sensakara, have suggested that it's good to start combining this knowledge and produce, let's say, nomograms or other type of instruments, either visual instruments or sometimes small calculators that you can find even on websites, where by just inputting this data, you can actually get a good prediction. However, despite all these things, we know, because we're practicing specialists, that sometimes things do not go as planned. And that's why, close to 15 years ago, it's a bit less, Renato Fansen's group, I think he was one of the first ones to introduce the concept of follicular output rate, FORT. And what does this mean? Again, it's been touched on by previous speakers. It is that scenario where you see a good number of antral follicles or a relatively good AMH, you stimulate with an adequate dose of FSH most of the times, but you still see that those follicles do not respond. And essentially you were expecting, for example, 8 or 10, and you just get 3 large follicles. And that's obviously in stark contrast when you do get at least 80% or even more of the follicles you expected. A few years after Renato Fansen's suggestion, the Poseidon group, Carlo Alvigi and colleagues, came to refine that concept and to add more context by proposing the follicle to oocyte index ratio, where essentially you have a good number of follicles developing, but then you don't end up with as many eggs. And they defined the normal FOI as more than 50% and an abnormal one as less than 50%. So these are distinct cases where we see that despite the fact that we expect things to pan out in a specific way, things do not. And that's very, very disappointing. For everyone who has to go to their patient who just woke up from the egg collection and say, I know I told you that we're looking likely at around 10 eggs, but we were able to get just 3. That's a devastating outcome, and it's devastating news for the patient. And the same applies, obviously, for the scenarios where, you suggested that we will have no problem reaching 15 eggs for you, and probably you'll never need another egg collection in your life. And then you end up just with 6 eggs, and all of a sudden, a big spiraling thing starts. So Carlo Alvigi and colleagues, in a very, very nice review on the FOI, suggested that among obviously the obvious culprits of, iatrogenic understimulation, having started with a very low gonadotrophin starting dose, or certain characteristics that have to do with how asynchronous the cohort was, or of course technical issues during the egg collection. Many times whenever the trainees in our department do not perform well, we'll sit and observe and see how well they've been trained in doing egg collection. Because we've seen, and this is actually supported by the literature, that the operator who performs the egg collection can be a significant factor on whether you obtain all the eggs you were expecting. But probably one of the most interesting bits was the fact that genetics might actually play a role. And that allows me now to come more to the core of my presentation. So this is a very simple slide of a very complex interplay between the 2 cells and 2 gonadotrophins that rule reproductive function. And this already has been discussed again in the 2 lectures before me, so I won't stay here. But essentially it shows that we need both FSH and LH and, of course, the steroidogenic products they produce to have a normal function of the follicular apparatus and a normal development of the oocyte. And if we look at the genomic or genetic aspect of things, you'll see that there are many different genes that have been identified to play a role both in the gonadotrophin independent and in the gonadotrophin dependent phase. And you can see here some genes that probably you would all imagine would be important, like AMH receptor, FSH receptor, LH-ACG receptor, BMP15, estrogen receptor 1, GDF, growth differentiation factor 9. So all these things, if you go to your basic reproductive physiology, they come up again and again as key contributors and key moderators of folliculogenesis and generally of reproductive function. So within the next 10 minutes, I'll try and review, do a very quick review of the basic genetic factors that have been studied by many colleagues, Manuela Simoni and others, Livio Casarini and of course Alessandro Conforti are key contributors in this area. We all know the FSH receptor, and again I won't stay here because it's not as important, but many polymorphisms, SNPs, single nucleotide polymorphisms, have been investigated as potentially modulating or moderating factors in the hyposensitivity to FSH or potentially hypersensitivity to FSH. One of the most common ones, or one of the most dominant in the literature, is the RS6166, which essentially is the replacement in in the area 680 of the protein of the amino acid asparagine by serine. It's, as I said, the most studied variant. It comes from a single substitution of a base from adenine to guanine, and it has been found, but again, this depends on the population. There are ethnic variabilities between population. The homozygous type of serine has been found to be around 15%. 40 to 45% is the asparagine, and the other are some heterozygous state. And it has been suggested that when you do have, like the variant of serine, particularly the homozygotes, that leads to reduced sensitivity, and that potentially can impact the results of ovarian stimulation. There are studies that have suggested that in these populations we need higher gonadotrophin doses to get equivalent results. This is the latest or the most recent meta-analysis on this specific mutation coming from Dimitri Loutradi's group in Athens. And in this systematic review and meta-analysis, they found, as expected, lower serum peak levels and estradiol levels with serine polymorphism compared to all the others. Again, suggesting potentially a hyposensitivity or functionality of the FSH receptor. The previous very, very wide systematic review and meta-analysis published in Human Reproduction Update by Carlo Alvigi shows that in terms of oocytes retrieved, we can actually have a statistically significant effect. As you can see here, if we compare the fully wild type with the standard type, we have about a difference of about 1 oocyte plus 0.84. And when you compare the heterozygous state with AA, there is no statistically significant difference. And when again you compare GG with AG, once again the GG genotype is the one that will lead to fewer oocytes. So as you can see, it appears that the GG, which translates to serine genotype, actually leads to fewer oocytes retrieved. This is a very interesting study, and these are the studies that we want to see whenever we have this type of observational evidence, where you can see we have group 1, which is serine, which was stimulated with 150 units per day. And then we have group 2, which is again serine, but this time they decided to start with a higher dose of 225. And you can already see the differences in terms of estradiol. When we have the asparagine, just with 150, you get even a higher response. You can see how the defect between group 1 and 3 can be almost completely mitigated by just increasing the dose. And even more revealing might be the case with the number of retrieved oocytes. You can see here that there is a difference between around 1.5 oocytes between the group 1 and group 3. But when you take serine and you just increase the dose by 50% in this case, you actually manage to completely negate the negative effect of genetics. And that's why I'm saying that this is an important study, because this is a randomized study and it proves that a pharmacodynamic approach can many times provide the outcomes that we want to see. Nikos Polizos, and this is part of a very big project he did with Lan Vuong from Vietnam, recently also offered some observational but still very, very useful data. In this case, they took around 368 patients, the majority coming from Asia. And that's important because, as I discussed, we always need to have in mind that ethnic groups might differ not just in the prevalence of this type of SNPs, but also on how important they might be for the function and what type of functional translation they might have in actual results. And essentially what they saw is that they found a higher prevalence of hyporesponders in patients with AG genotype, but that was about it. There were some statistical differences, but they seemed to be of limited clinical value, according to Polizos and colleagues. At the same time, in that same, as I said, very interesting big paper, observational study, they studied other SNPs of FSH receptor. This is a different one. This is the RS6165. And again, a higher prevalence of this type of genotype in hyporesponders, and some statistical differences, but again, of limited clinical value, and importantly, not very consistent with each other. Another type that was also studied by Nikos Polizos, again, similar findings. Yes, you could sometimes find statistical significant differences in some of the outcomes examined, but even the authors themselves understand that this is probably of limited clinical value. We have other FSH-related polymorphisms, like the FSH-B gene promoter, and this is also something that has been studied, not to the same extent as the RS6166, but it actually leads to reduced FSH secretion, and that has been hypothesized that it could impair follicle recruitment. However, what it also has been shown is that potentially affects the FSH-LH ratio, and it might predispose to PCOS. But a consensus that was authored by many of colleagues present in the room actually came to the conclusion that probably because of contradictory data, we still do not have strong evidence that it could be influencing oocyte yield and baseline FSH. We talked about 2 cells and 2 gonadotropins. So what about LH? And we heard Livio Casarini's talk about how important LH receptor can be. So we also have some polymorphisms in LH that have been studied, understandably. And probably one of the most important ones is the serine 320 asparagine. And this is a very interesting data coming from India. Why? Because it seems to align well with what we just heard from Livio Casarini. Essentially, they took 2 groups, and this was not randomized, but it was interventional. And in group 1, they did supplement from day 6 with recombinant LH, assuming patients had shown that their LH, endogenous LH levels had dropped. Whereas in group 2, they actually performed a genetic assessment based on this specific allele and polymorphism, and did a very tailored, patient-tailored and genomic-tailored supplementation of LH. And the interesting graph is here. You can see that group 1 had recently, relatively decent pregnancy rates, which were relatively consistent across the genotypes, but the serine genotype did far worse, even by adding LH from day 6. Whereas in group 2, where LH was supplemented from day 1 using this specific scheme, you can see that the live birth rates were entirely comparable and, interestingly, much higher than what had happened in group 1. And I think these type of studies, are great because offer just food for thought. Estrogen receptors, we know how important is the estrogen. A couple of polymorphisms have been identified as being the most important. Usually we do have conflicting evidence, unfortunately, here as well. Here you can see two studies, one from 2007 and 2014, which show some potential, depending on what type of genotype you have, that you might respond differently in terms of follicles and oocytes. And here's another study using another genotype, another SNP, which again shows some trends or some potential for having an effect on the output. However, other studies, which are too many to mention, actually have failed to detect similar associations. Therefore, despite these potentially interesting results on the effect of estrogen receptors, I think that more work is needed. AMH and AMH receptor polymorphisms also very important and have contributed, and it has been suggested that specific polymorphisms might actually be associated with poor responders. These are the latest data by meta-analysis by Chen and colleagues, and you can see here that more oocytes were retrieved when you had this specific type of genotype compared to the TT genotype, and for the receptor, they weren't able to find any differences in oocytes retrieved with the genotypes that felt that would be affecting, the outcome. So not really that much of encouraging data for AMH. But of course, these are all single gene studies, and that's always, whenever you sit down with a molecular geneticist or with a clinical geneticist, they'll say, but Christos, this is not how things work. We have gene-to-gene interactions, we have environment-to-gene interactions, and in most cases, you need to do like a GWS. You need to do a genomic-wide analysis association study to know what's really happening. Unfortunately, in our field, the only such study came from, that I could find at least, and I'm happy to be corrected if there are more, which I might have missed, comes from Bart Fauser group, and it's already quite old, it's almost 15 years old, where they couldn't identify a specific combination of SNPs that was predictive of ovarian response and other markers. But it's important to note that that was only in 102 patients. And that might be very, very low, very underpowered to detect meaningful associations. Finally, and this is my last slide, I would be remiss if I didn't mention this. Why? Because many have read the case reports. So there are some genetics where some patients might be susceptible to OHSS. We've seen OHSS spontaneously in spontaneously conceived pregnancies. So there has been certain data on very rare FSH receptor mutations that might lead to overactivation and continuous activation, or even in polymorphisms in VEGF, which lead to a clinical picture similar to OHSS. But again, this is early data, and probably we need to wait for more and more data in order to see whether we can identify patients that are at particular risk of OHSS even without being hyperstimulated. So, concluding, what does the future hold? So we already have started seeing what the future might look like, and that comes with the power of like machine learning and getting more into data-savvy, techniques of analysis. This is a really nice study from Zielinski and colleagues where they looked at just 14 genes of interest and used gradient boost machine learning. And you can see that AMH, of course, in terms of prediction of two oocytes, had the highest strength, and AFC, but then specific genes also showed up. Of course, they didn't have the same effect. This is from the same study. You can see that most of the variance in the prediction of the ovarian response still comes from, the regular AMH or AFC, but still, it wasn't insignificant. They could add meaningfully, and having genetic data could predict and could upgrade the personalized prediction of ovarian response. This is another study also following the same pathway using machine learning, and in this case they investigated 31 polymorphisms, and again they identified 6 genetic polymorphisms that could help refine our prediction models. They weren't like the most important predictive factors, but generally could help improve it. So The ideal scenario is you have a case like Helen here, which she's 32, AMH 1.6, and she has the serine RS6166 mutation. And as we've shown, even with a randomized controlled trial, if you knew that beforehand, rather than having to face the surprise of hyper-response, you can increase the dose. Or in other cases, you might have scenarios where by adding LH, you could also improve the outcomes for certain carriers of specific polymorphisms in the LH receptor. There are some limitations that I need to mention. Obviously, when we try to personalize genetically, we need to be always very, very cautious. Why? Because of the multifactorial nature of what we're trying to do. Ovarian response is essentially polygenic, and that means that you cannot have just one or two genes, and there are other environmental factors that might also play a role. The effect sizes of the most common variants we have identified in the literature are actually pretty modest, and we've seen that in the latest studies that I showed you just earlier. Also, combining and comparing different studies that have looked in genetics can be a challenge, and I wouldn't recommend it for someone who's not very methodologically sound and at the same time understands the technologies. And last but not least, we have ethical and practical issues which come about, genetic testing costs, who's going to pay that, patient consent, where are we going to store the genetic and genomic data, and all these things. And importantly, if we were to do this, patients might get inflated expectations and might think that we now have the complete picture and we can predict exactly what's happening. And that's unfortunately not the case. So polygenic scores might be the future. Of course, we have some early data in certain labs of the world suggesting that they can do a good job and potentially better job than single gene variants. But we still need to integrate these with clinical factors and always have in mind that feasibility is going to play a big role. So we need more and more data, and we need these type of tests. Clinical relevance is still modest. It's very important for the ones that actually have these genes in many, many cases, but at the population level might not be as important. Of course, there is a huge upside in terms of the potential we have for truly individualized and personalized pharmacogenomic approaches, but we always need to have different people working together, reproductive endocrinologists, geneticists, big data scientists, in order to get the best out of the data. So what does the future hold? Well, it's going to be bright, and I hope you share the optimism with me, but as with any source of bright lights, always make sure you bring your shades. Thank you.